Good afternoon, ladies and gentlemen. Welcome to Academia Sinica special lecture today. We are very honored to have Dr. Martin Chalfie as the speaker. Now, let's welcome our Vice President, Mei Zhou, to give us an introduction to Dr. Martin Chalfie.
Okay, ladies and gentlemen, good afternoon. It is my honor to welcome everybody to this special Academic Sinica lecture to be delivered by Dr. Martin Chalfie in a few minutes. President James Liao had planned to personally host this lecture, but he's sending his regrets because he has to attend a meeting called by the president of the country. Therefore, on behalf of Academia Sinica and President Liao, I will extend our sincere gratitude to Professor Martin Chalfie for visiting our campus and for delivering this special lecture. Okay. And Professor Chalfie was educated at Harvard University, earning his bachelor degrees in biochemistry in 1969 and a PhD in neurobiology in 1976. He currently holds the prestigious title of university professor at Columbia University. Among his many honors, he was awarded the Nobel Prize in Chemistry in 2008 for his groundbreaking work on the discovery and development of the green fluorescence protein, which was originally found in jellyfish. Okay. His pioneering research in the late 20th century using the tiny transparent roundworm, C. elegans, profoundly advanced our understanding of the nervous system. His innovative approach involved using the GAP gene as the genetic marker, which allowed us to visualize and track the activities of specific cells and molecules within living organisms. This work illuminated the inner workings of the individual nerve cells and produced a major impact on fields such as developmental biology, neurology, and cancer research. Professor Chalfie is a distinguished member of the U.S. National Academies of Sciences and also Medicine, as well as a foreign member of the Royal Society. He has served as president of the Society for Developmental Biology and the American Society for Cell Biology. In addition, he chaired the Committee on Human Rights of the National Academies of Sciences, Engineering, and Medicine. Okay. So finally, I would also like to express our gratitude to the Foundation for the Advancement of Outstanding Scholarship and the CTCI Foundation for generously sponsoring today's lecture. Okay. So we'll very much look forward to hearing his enlightening and also thought-provoking lecture entitled The Continued Need for Useless Knowledge. Okay, before Professor Chalfie begins his lecture, I would like to invite him to come forward to receive a special gift as a token of our appreciation for his role as the Academic Sinica lecturer for 2024. So please join me in extending a warm welcome to Professor Chalfie.
Thank you very much for that very kind introduction. And hopefully there it is. There's the first slide. This is a very general talk. It's a rather unusual title, I agree, for a scientific talk, The Continuing Need for Useless Knowledge. So let me explain that right from the beginning. It's actually taken, or at least the words useless knowledge, are taken from a very interesting magazine article that was written in 1939 by this man, Abraham Flexner. His article was entitled The Usefulness of Useless Knowledge, and I'm just continuing that. But as you can see from the quote at the bottom of the slide, he said, intellectual life is on the surface a useless form of activity. But the pursuit of these useless satisfactions proves unexpectedly the source from which undreamed of utility is derived. And what he really is talking about throughout this article is the importance of basic research or foundational research and why that is important. But he starts off in a wonderfully interesting way in this article. And he says that he is talking to the industrialists. Whoops, where is he? He's not there yet. Should have been there. Something's wrong with the slide. We'll put both of them up. He's talking to the industrialist George Eastman, who started the Eastman Kodak Film Company. And he says to Eastman, who do you think is the most important scientist in existence now? And Eastman, without any hesitation, says, oh, it's Marconi. Clearly Marconi, because Marconi came up with wireless and radio. That's the most important person here. And then Flexner. I should explain a little bit about Flexner. If you're a pre-med student in the United States, you have to take a lot of college courses before you can go to medical school. You have to take biology and physics and chemistry, organic chemistry. You have to take math, all sorts of, all because of Flexner. He was in charge of the committee in the early 1900s that made the rules of what, so he's ruined the lives of potential medical students for over 100 years. And then he wrote this article. And I think he's very determined. He says to Eastman, after Eastman says this, he says, you're wrong. This is not a good person. This is not the best scientist. In fact, Marconi's share was practically negligible. He was inevitable. If it wasn't him, it'd be somebody else that would invent radio or wireless. The really important people were the people that developed the theory about electromagnetic waves and to understand them. And those people were Clerk Maxwell and Heinrich Hertz. Without them, nothing would have been made. And he says about them that Hertz and Maxwell were geniuses without thought of use. They weren't looking for the applications. Marconi was a clever inventor with no thought other than use. Now, frankly, I think he's a little unfair. Because you need both. You need to have the basic knowledge, and you need to have people using that basic knowledge. They really come together. So I would really say that you need both new knowledge and new applications. The problem comes in, I believe, when people say, don't we have enough knowledge? Can't we use everything we already know to apply to some problem? And the answer, unfortunately, is we need to learn a lot more. And there's a couple of other examples I have of this. I'm going to give you a couple of examples. One from a non-scientist, the author Mark Twain, in a letter in 1903, even before this, he says, it takes 1,000 men to invent a telegraph, or a steam engine, or a photograph, or a telephone, or any other important thing, and the last man gets all the credit, and we forget the others. to bring this home maybe a little bit more, I want to show you an infographic made by the Quartzoff company. It's impossible to see, but I'm going to show you the entire infographic. Here it is. You don't know what that is, but if you look at, if you go to that website, the quartzoff.com website, you can see the whole thing and look at it. But what is it about? Well, if we look up at the very top, how did we get the iPhone? What were all the previous applications? What was all the previous knowledge that was needed to produce a smartphone? And as you go down the chart, if you go down a little bit further, you see all of these contributions to the camera, to the Wi-Fi, to the every other aspect of the computer and everything else in the smartphone, and all of these, and this only goes down to the middle 1800s. There's a lot more of the chart there, and when you get finally down to the bottom, you've got the oldest lens in the world, 750 years, 750 BCE. You have Euclid's optics at 300. You have the discovery of static electricity, and so on. All of these things are what are built upon to finally get to this final product. It's not the last person. It's everything building up to it. And I want to give one more example, because I like giving a lot of examples, but this is one of my favorites. It's a little outdated, but it's a short video that I hope you can hear and see that was put together by the people at the bottom of the slide. These are graduate students and postdocs at University of California in San Francisco who entered this video and won a contest by the Federation of American Societies of Experimental Biology. They wanted to have a video that would explain the importance of basic research. And so this is the Stand Up for Science, and this is the winner. So let me just play this for you. You'll understand what it is, I hope. Oh no, the sound is not here. Is there anybody that knows how? I could say what the sound is. I've heard this so many times. So let me explain. They say they set up a stand. This is horrible. They set up a stand and asked people, which would you do? It's 1960. Would you pay for money to find out how bacteria protect themselves, or do you want money to find a cure for insulin, for diabetes? And everybody says, no, we want to cure diabetes. We don't want to do that. And then they say, what actually happened? NSF and NIH actually supported research into how bacteria protect themselves. And what they found is that when a bacterium is infected by a virus, the virus comes in. Eventually, they'll show a picture of the virus infecting the cell. It brings its DNA in and incorporates its DNA along with the bacterial DNA. And that bacteria can prevent this takeover by the virus by having enzymes, which we call restriction enzymes, that cut up the viral DNA and thus protect the virus, the bacterium from infection. And that these same cutting enzymes can be used to cut human DNA, put that into bacteria, and now that DNA can encode different human proteins or anything else that you want to put into it, and make a lot of it because bacteria are really good at that. And so they put in the gene for human insulin, and it could make lots of human insulin, and it was clean and good and everything. And they point out here that before this, it was really expensive because insulin was derived from fetal cows, and they'll say that the first person that was given it got really sick. But now they have insulin that's here as well, and has produced a treatment for diabetes. And in addition to that, this basic technique has been used all over the world to do so many other things, to increase our knowledge, to get more information, to answer questions. And here are some of the examples that they're giving of all the things that this one discovery has enabled. And they say, who knows how much more can be learned? And then they give other things that have come out of basic research. And you'll see they cross out all of them but one. And then they talk about the continuing need for this and the fact that we need people's support for this to be able to keep the discoveries coming. And they point out that it's everybody that is contributing to this, and it allowed for the discovery of something that cuts DNA, and there will be other things in the future. And then I think they have the credits where they show everybody's picture and all these people these are the people that made the film, and they're saying this is important. And I don't know about George, but he's part of the picture nonetheless. So you get the idea of the importance of basic research. You don't really know what's going to happen. There's a famous story about Benjamin Franklin that after the American War of Independence, he was the first ambassador to France. He goes to France in the 1780s, and he sees the Montgolfier brothers' hot air balloon going up. He's amazed at it. There's a person in the crowd that says to him, what's the point? What's the use of this thing? And he's reported to have turned to the person and said, what's the use of a newborn baby? So you never know what's going to develop for a person or science or whatever. In the United States, there's actually a prize for useless knowledge. This was started about 10 years or so ago. It was put together by the American Association for the Advancement of Science, but also 4 Congress people, 2 Republicans, 2 Democrats, very fair, and it's called the Golden Goose Award. And the Golden Goose Award is the award highlights and honors examples of scientific studies or research that may have seemed obscure, sounded funny, or for which the results were totally unforeseen at the outset, but which ultimately led, often serendipitously, to major breakthroughs that have had significant societal impact. So what are the things that they've given? Well, I'm very proud that I was in the group of first people that got this, because they gave it to green fluorescent protein, but they also gave it to Charles Towns for developing the maser, which then led to the laser. And the other group was the people that discovered that coral, dried out, turns out to be a spectacular substitute for bone during surgery. And there are other things here, but I want to point out one example, which I'm very impressed with, and that's the one at the bottom about rat and infant massage. It turns out that there were a group of researchers, and they were making a particular, I think, rat mutant. They were very concerned about keeping these animals alive, and they were a little worried that after the babies were born, the mothers might reject them or have a problem with them, or they would be at risk. So they decided that they were going to take the baby rats away from the mother and put them in the wonderful environments and make sure everything was right. And to their horror, they found that the babies did not do well at all. So the next brood comes up. They decide, we're not going to take them away. We're going to see what the mother does. And what they found the mother did, and the babies thrived. And the babies thrived, in part, they realized, because they watched the mothers, and the mothers are constantly licking the babies. So they wonder if that's important, so they do an experiment. They take the babies, they didn't lick them. But they take a swab and they stroke the babies. And the ones that were stroked, that had that touch, they thrived. The ones that didn't, did not. And they realized that a very important aspect of the development of these newborn rat babies was, in fact, mechanically driven. That the sense of touch was very important, and the application of a touch stimulus was very supportive. Very interesting discovery, but then people realized maybe we should be doing this with human babies. Maybe with premature babies, touch is very important to have them thrive. And apparently all over the world, this is now done for premature babies. So a wonderful discovery, not what they were looking for, but a very basic understanding that led to a very significant health increase. Now, I think new knowledge always allows for new approaches. And I think it's quite understandable if when people have a disease themselves or a relative is afflicted by a disease, there's a very strong desire to get a solution, to find a cure or a treatment. It's horrible to see someone suffering. And so I think it's very understandable that disease-oriented advocates would want sort of an immediate answer. But I think that's very hard, and I think biologists have made a good argument over the years that in addition to trying to directly answer a question, that we also need to build up the body of basic knowledge so we can have new approaches, whether it's CRISPR-Cas9 or RNA interference or some other discovery of a new enzyme that can maybe be inhibited or whatever. And so I think we've been able to make a good argument, and I think many disease advocates, I think, recognize that we need to have all of this resting on a foundation of basic research. But I wonder if we shouldn't be making this argument more generally. So again, I don't know what it's like here, but in the United States, if you turn on the television and hear anything about climate change, what you hear about is clean energy, not using fossil fuels, all sorts of engineering solutions, carbon sequestration, all sorts of engineering solutions to the problem. I have a good friend, Manu Prakash, who's at Stanford, who studies this, and he says, you know, we have absolutely no idea how vast bodies of water, like the oceans, cope with increased CO2. What is actually happening there? We don't know the basic science behind how the world copes with these various changes that are there. Again, large-scale ocean activity of microbes. We just don't know the information. So I would advocate that you need to start saying the same things I said about disease for climate change and getting support for this, or food security, or any of a number of things. We need to have the foundational work to increase these. So I want to switch a little bit here to talk about how does one make discoveries. We need this basic research. How do we make them? And there's a wonderful quote by the physicist Enrico Fermi that goes, if the result of your experiment confirms your hypothesis, then you've made a measurement. If the result is contrary to the hypothesis, then you've made a discovery. So we often teach the scientific method. We say, you know, you have to come up with a problem, then you have to have a hypothesis, and then you test the hypothesis and everything. I think it's the other way around. I think you make an observation that may be quite accidental, and then you ask questions. Why? How does that come about? So I'm going to talk a little bit about methods that might address this, but before I do that, I want to give some examples of people that have made accidental discoveries that have led to their getting a Nobel Prize. And the first example I want to give is Robert Wilson and Arno Penzias, who were at Bell Laboratories in New Jersey in the United States. And Robert Wilson, his PhD thesis, the experiment he tried to do for that didn't really work. And I think he was still feeling bad of it when he was at the Bell Lab. And they discovered that there was this 30-foot microwave telescope that somebody else had built. And it was just sitting there. And Robert Wilson decided, I can go back to that problem I never solved and see if this will help. Now, he actually never did that because when they tried to calibrate the machine, they did so by pointing it at the Milky Way, where they thought they'd get a great signal, and they did. And then they pointed it 180 degrees away from the Milky Way. And what they wanted was it to go to zero, and it didn't. And they went through a whole series of, was it electrical lines near the telescope? No, it wasn't. Was it the pigeons that were nesting in the telescope? No, it wasn't. They eliminated everything until they finally realized it can't go to zero because it's measuring the background radiation in the cosmos, which turned out to be the best evidence that there had been the Big Bang. And that's what they won their prize for. So they didn't even get a chance to calibrate their instrument when they made it. Another person that won a Nobel Prize, John O'Keefe, in his Nobel Prize speech, says that one day he was doing electrophysiology in the lab where he was sticking the electrodes into the thalamus to measure some activity, when by mistake he put the electrode in the wrong place. And when he did, he discovered a cell that responded to place, where the animal was in space. And that was the discovery of place cells, which he won the Nobel Prize for in 2014. So sticking the electrode in the wrong place. My next example is the one closest to home, Osamu Shimomura, who shared the GFP prize with me. And Osama was an amazing biochemist, very, very talented, an astonishing guy. But when he started to work, I want to give an advertisement here. His daughter and son-in-law convinced him to write a memoir. It's called Luminous Pursuit. And if you have a chance, read it, because his life is really quite amazing. As a teenager at 16, he's told, you can't be in high school anymore. You have to work in a factory. You have to quit and work in a factory. So he does that. Turns out to be a good thing that happened because it was 1945 in Nagasaki, Japan, and by being in the factory, he was protected when the atomic bomb destroyed the city. He writes about how he walked the 10 miles home, or whatever it was to his home, while there was this rain that had ash, radioactive fallout and everything. And he said his white shirt turned gray. He gets home, his grandmother takes one look at him and basically says, take a bath, get out of this oven, and he attributes that to saving his life from radiation poison. He can't go back to school after World War II. He eventually, he gets his high school degree, but the college has been destroyed. So he goes to the only college available to him, and one he said he wasn't really interested in, which was the pharmacy school. Nothing against pharmacy school, he just wasn't that interested in it. He graduates, he goes to work as a technician in a lab at Nagoya University, and he makes a wonderful, somewhat accidental discovery, but realizes what he's done, and as a result, he gets invited to come to the United States to do more research on a fundamental biological problem. How is it that different organisms can generate light? And it turns out that there's lots of organisms that can generate light, and they all do it differently. So fireflies are different from glowworms, that are different from bacteria or fungi. They all do it differently. And he is invited to come, among other things, and study why this jellyfish produces this green light. His boss at Nagoya University does something quite unusual. He realizes that if Shimomura just goes to the United States, he won't be paid as much as if he went with a PhD. So he arranges as a going-away present that the university would give him his PhD. Not a bad present. And he comes to the United States, works at Friday Harbor Lab in Washington State on the jellyfish, and fails completely to be able to figure out the biochemistry. It doesn't work. He does experiment after experiment after experiment. He bribes his kids to bring him more jellyfish, and he just keeps trying, grinds things up. Nothing works. The whole summer, it's a failure. At the end, sort of at the end of the summer, one night, he's been working late into the night. It's already dark outside. I think he wanted to go home and have dinner, and the experiment has failed. He takes the prep and throws it away. He's cleaning up. He throws it away into the sink, which had some seawater and a couple of other things in it. And he's about to leave. He turns off the light and happens to look back at the sink and sees that it's glowing. That's a real surprise. Thinks about it for a little while and realizes that maybe there was something in the seawater that he never added to the solutions before. Thinks about that for a while and says, seawater has calcium ions. And sure enough, when he repeats the experiment, every time he squirts in a solution of calcium ions, he gets a flash of light. And he uses that to purify the protein, which he names after the jellyfish. For any students here, I just want to tell you that this is sort of a type of scientific method. If your experiments don't work, throw things in the sink. Throw them on the bench, throw them on the floor, pick them up, and then try all over again. It seems to be, it works for some people. I don't know if it's the thing you should do. In any case, what he finds is this protein that he names after the jellyfish, he names it a corn, plus calcium will produce light. So he has solved the problem, sort of, because he realizes it's the wrong color. So another accidental discovery, the wrong color. He thinks about that for a while. He squirts calcium into all his other samples. Nothing happens. So he finally says, maybe there's a different protein that converts light, that if you shine blue light onto it, it'll make green light. So he takes the handheld lamp and he looks at all the samples and finds that sure enough, there's another one. In his 1962 paper announcing the purification of a corn, he writes a footnote saying, look, there's this other protein. If you shine blue light on it, you get green. I'm going to call it the green protein. Of course, we don't call it that now. We call it green fluorescent protein. And it is the thing that's needed to allow for the production of the green light. I just want to point out that, as far as I know, this is the only footnote that won its author a Nobel Prize. So you don't have to write a lot for this. Now, I heard about this molecule in a seminar in 1989. I worked on a transparent animal, and so the idea that there was a protein that you could activate, not chemically or any other way, but just by shining blue light on it, and then you'd get green light coming out. I got really excited about this. I thought this was fantastic that we could do this. And that, I just want to point out something about this molecule. This is the most astonishing looking molecule in the world. It is a cylinder with a single strand going up the middle, of which the important part is a rearrangement right there. It looks exactly like it's supposed to. It is a lantern. And I'm saying, I could put this in my animal that's transparent. This will be fantastic. And so we did this. Douglas Prasher as a collaborator who got the DNA for this. Gia Skirkin was an undergraduate. I should explain my color code. You may notice that, well, anyone from my lab, their name is in red. Anyone who is a collaborator, their name is in blue. And anyone whose name is in black did something I wish I had done. Okay? So they were able to put, Gia was able to put it immediately into bacteria, showing that it could be used as a biological marker. And then we showed we could put it in the worms that we study, and we could see the cells that we like to look at, and other people put it in fruit flies and zebrafish and mice and all sorts of different organisms. And because it's so small and diffuses throughout the cell, you can see the entire outline of the cell. For example, here's a picture of a Purkinje cell, completely outlined. So one can see the structure. But the real plus of this molecule is that all you have to do to see it is to shine blue light on it. And that means you can see it in a living organism. And I want to show you two very nice movies. They're both of the early nuclear divisions in the developing fruit fly embryo. And they were both done by a woman named Rosalind Silverman Gavrila, who I've never met. She made these movies many years ago as a graduate student in Canada. The movie on the left, you know that when a cell divides, the chromosomes are separated by what's called the spindle. And so one of the spindle proteins has GFP in this movie on the left, and I'll let you see it. It's very much sped up, so you can see 4 rounds of division. You see the spindle forming there. Division's complete. We'll go through another round right now, another one here, and it will cycle through these. And as you look at this, I think it's inevitable to say, wait a minute, everything's happening at the same time. In fact, she named this movie In Synchrony. And that's one of the advantages of seeing a preparation live. You get a chance to see what's happening over time. You get a dynamic view, not a snapshot view of what's going on. And it immediately invites the question, how is that possible? And we do know a lot about the control of cell division and what allows this to happen. On the right-hand side is a falsely colored image. It's falsely colored. The convention is the more you have of whatever you're looking at, that's going to be colored red, a little bit less orange, and go down the rainbow until the least is blue. So you can see that there's some cells that are sort of blue here, green, and these are more. So there's a slight difference in the amount. This is GFP that has attached to it a small peptide called the nuclear localization signal. And this peptide, if it's round, whatever it's attached to, gets sucked right into the nucleus. And so we'll watch 4 more divisions of this. By the way, she names this after the Van Gogh painting Starry Night, and I think you'll see why in a moment. The nuclei break down, now it's all over the embryo. The nuclei reform and it gets sucked back into them, and that really does look like the Starry Night picture. They break down again. But what you'll notice here is it's not synchronous. It's going from the lower right-hand corner to the upper left-hand corner. And we believe, or I believe from talking to people that do similar experiments, that her thumb was maybe a little heavy when she made this preparation. So it's a very sensitive way of looking for damage in here. But again, to be able to watch what is actually happening over time is really quite spectacular. Now, I feel that all discoveries, whether you talk about the laser or whether you talk about GFP, these things lead to more fundamental research. And one of my favorite examples of this is the discovery by Tony Hyman and Cliff Brangwyn of these new components in the cytoplasm, these phase-separated particles. Right now, this is one of the hottest areas of cell biology, and they were only seen because they were able to be labeled by GFP proteins that happen to be part of them. It's a wonderful discovery. I'll note it came after the Nobel Prize in this, but another example, and this is more applied, is to ask the question about viral infection. When we teach to undergraduates about viruses, we usually talk about bacterial viruses, and we usually say that what happens is that when the viruses grow within a bacterium, the bacterium explodes, the viruses go everywhere, and they infect other cells. And so the question is, what about the AIDS virus? Does that do that, or does that do it all the time? Because if the virus is spewing out of cells, maybe antibodies could grab onto it and control it. But what actually happens? So these researchers, Hubner et al., decided to make HIV, the virus that causes AIDS, and put it into a mouse cell, and here's a viral particle, and you saw it going into another cell. No explosion. It transferred from one cell to another, immediately inviting the question, how can this be controlled? How do we think about this? So watching something with maybe a very direct application. Now, I think all of this, having both of these things, sometimes we think about this as fundamental research gives rise to applied research. I think this is more circular. It goes in really, each one helps the other and is here. This also happens with the laser. There have been enormous fundamental discoveries enabled by the laser. Low temperature physics, gravity waves, all sorts of wonderful things. In addition to developing CDs and DVDs and the scanners at the grocery store that allow you to get out faster, and laser surgery. So they're all there, and each thing sort of helps one another. But I think in addition to this, there are really the unexpected things. So I want to give you a couple of examples of things that I find surprising and unexpected. So there's going to be 2 slides, I think, of this. One of them, let me talk about the work by Jeffrey Waldo, who is at Los Alamos Lab in the United States. Jeff is interested in protein folding. How does a protein, as it's made off the ribosome as a linear chain of amino acids, how does it fold up? And is there a way, if a protein doesn't fold correctly, can we get it to fold correctly, to do the right thing? And so he had a wonderful idea for this thing of being able to look at protein folding. He made a fusion. The protein he was interested in, and GFP. And what he found is if this doesn't fold correctly, this won't fold correctly. And if this doesn't fold correctly, you can shine all the blue light you want on it and nothing will happen. if you mutate that protein so that it will fold correctly, that will allow the GFP to fold correctly. And now when you hit it with blue light, green light will come out of it. It's a wonderful adaptation of this. Another interesting and unexpected thing was work by Lynn Regan. She is or was the chair of the chemistry department at Yale, and she did an experiment that at first you're going to sound, that sounds a little silly. She took GFP and she cut it in half. Now, it may not be a surprise to you that if you cut GFP in half, it will not work. But what she did next was really brilliant. She attached to each half other proteins that are known to come together. And when these come together, they allow the two halves of GFP to come together, and all of a sudden it works. So what this is, you can put any two proteins you want here to test whether they will bring this together. And so you can test whether proteins are interacting with one another. It's a wonderful, wonderful technique. Other things that have happened in the GFP world, some unusual things. There's a couple of researchers at Agricultural Station in Japan who stuck GFP and other fluorescent proteins into silk moths and produced fluorescent silk. This is some of the clothes that have been made from this fluorescent silk. This is a nice idea, and I have a problem with it. And the problem is, it's so cool, you have to spend your entire life holding a UV lamp, walking around, showing people how interesting your clothes are. So maybe not as good as you want. Other people have used GFP to make cellular lasers. Bob Burlage, at the time at Oak Ridge National Lab, knew, I'm going to give you another example, he knew that there was a regulating element in E. coli, I think, that would turn on genes in the presence of the explosive TNT. So he made E. coli with that, but now they make GFP. Great. And he could sprinkle some TNT on a petri dish that he could get the bacteria to be green. And you might say, why did he do that? Well, he knew that landmines, probably one of the worst aspects of war because they kill people after the conflicts are over and they kill innocent people, landmines leak TNT. And so this was going to be a TNT detection system. And he had a friend bury 5 landmines in a 3 meter by 5 meter plot of land. They're smart people, they were disconnected, but they did have TNT in them. He sprayed that area with his bacteria and came back at night to take a picture of the fluorescing cells and was able to locate the landmines. This sounds great, but there is a problem. This is a type of experiment that you never want to give you the wrong information. You don't want false negatives. And so I know of several groups that are still working on perfecting this as a way of making sure that it's not going to hurt somebody to do this. But it's something that several groups have been working on. 2 other examples. There's an artist named Eduardo Koch. He's originally from Brazil, lives in Chicago, and he wanted to get people talking about art and science at his various art shows. How did he do it? He commissioned a French biotech company to make him a GFP bunny that was the family pet for many years, and that he would bring around to his art installations to get people talking about science and art and science and technology. And finally, GFP comes up in some very, very strange places. I would say that the first possible human transgenic is this guy. if you look at the beginning of the Ang Lee movie Hulk, you will see a jellyfish. And you'll see a hypodermic come in and get the GFP right out. Shimamura should have known that. And it's then obviously injected into Bruce Banner and probably gets into some gene in his skin, and that's the end of it. And he turns green whenever that gene gets turned on by anger. I actually know the screenwriter of this movie. His name is James Shamus. He's a professor at Columbia. His daughter and my daughter went to elementary school together. And after the movie came out and my lab forced me to go see it, that's really a lie. I wanted to see it anyway. I went up to him and I said, James, how do you know about my work? That's fantastic. And he said, I have no idea what you do. And I said, no, that GFP, how did that come into the movie? He said, oh, that. We had a student from MIT, and he forced us to do that. Fine. Okay. I don't know if this book exists here in Taiwan. It's a wonderful children's book in the United States called If You Give a Mouse a Cookie. I will tell you why I'm introducing it. This is a horrible book because the mouse comes in and says, I want a glass of milk. But if you give the mouse, I want a cookie, if you give the mouse a cookie, it asks for a glass of milk. And after you give it a glass of milk, it wants to have a straw. And then after it uses the straw, it might have some milk on its whiskers, so it asks for a napkin. And the book goes on and on and on and on. He's asking for more and more things, and at the very end, he asks again for a cookie. So, you know, it never ends. This is a perfect analogy to scientists, because you give people a perfectly good green fluorescent protein, and their first reaction is, can we get more? What about other colors? What other things can we get? And the person who really contributed this the most, or did it at first, was Roger Chen, who was the third person that shared the Nobel Prize. Roger was able to mutate GFP and immediately find a version that, instead of giving off green light, if you were activated by ultraviolet light, it gave off blue, so it's blue fluorescent protein, or BFP, and then other manipulations later led him to make a yellow-green version, which he called yellow, or YFP. But the problem with modifying GFP to get other colors reached a limit. So only a few colors were found. And it wasn't until two researchers in Russia came up with another idea. These people, Mikhail Matz, who's now in the United States at Austin, University of Texas in Austin, Sergei Lukyanov, who is the head of the medical school in Moscow, they knew, I think because they like to do scuba diving, that corals were wonderful sources of fluorescence. And so they asked the question, is the gene in the coral similar to the GFP gene? Can we use the GFP sequence to find a similar sequence in the coral? And they did, except that the corals were fluorescent not to produce green, they produced red. And as a result, they found the first red fluorescent protein called DS red. The DS refers to the species of the coral. And a lot of subsequent work, a lot of it by Roger's lab, led to a whole wealth of additional fluorescent proteins. In fact, there's a website called FP.org, something like that, fluorescentprotein.org, and it lists over 1,000 fluorescent protein variants that have been made now. But this fits the whole spectrum, and they got tired of letters, and so they started naming things after fruits. So there's tomato and orange and cherry and grape and plum and all of that. So they thought that was a little more fun, which it might be. But they were able to make all these wonderful pictures. So what happens when you have other colors? Well, one of the things that happened was, what if you take random expression of maybe 4 colors and have every cell have a different random expression? You can label each cell with a unique color. So you don't need all the colors, you just need to have maybe 4, and Josh Sains and Jeff Lichtman decided to do that. And they started off by expressing these in the nerve cells in mice, in the brains of the mice. And so if you're going to put all the colors of the spectrum into mice in their central nervous system, I think they came up with the only name one would want to use, which is brainbow. And these are some of their pictures. But another thing, we use it in my lab to look for mutants because we're interested in a set of nerve cells. Maybe a little hard to see. That black arrow is pointing to a cutoff end of a nerve cell, a little green dot. And as the animal gets older, the skin, the blue, completely encases it, pulling it away from the red, which is the muscle. So when we put RFP in the muscle and GFP in the nerve cell, we see this nice gap, except in mutants where it doesn't occur. And we've now found 5 different genes that have this pattern, and 18 others that have a partial pattern. So we're able to find mutants on looking at an activity that we would never, ever be able to study. Roger championed another use for GFP, which involves something called FRET, for Forster resonance energy transfer, sometimes fluorescence resonance energy transfer. And the way this works is you need 2 different fluorescent molecules. If the 2 molecules are apart from one another, if you use ultraviolet light to activate the protein, most of the time you'll just get blue light out. Some of that blue light may make it over to the yellow fluorescent protein, and you'll get a little yellow out. But basically, you don't have it. But if the 2 molecules are absolutely touching one another, they have to be very, very close. And that happens in this case by calcium binding to the middle part, bringing the 2 parts together. Now, when the ultraviolet light activates the blue fluorescent protein, instead of making blue light, it transfers its energy to the yellow fluorescent protein, and now you get much more yellow. So simply by looking at the ratio of blue to yellow, one can decide how much calcium there is around. It's a wonderful tool because just like GFP, it's a protein. It's encoded by DNA. It can go from one organism to its progeny, to their progeny, keep going. And there have been there's several hundred of these biological monitors that have been made this way, looking at enzymatic activity, or looking at redox, or looking at pH, or looking at a number of different features in the cell. And one can look, again, just like with GFP, in living cells and looking at changes as these take place. And finally, one difference between all the men in this room and all the women in this room is that women have, as female mammals do all over, they have 2 X chromosomes, and one of the X chromosomes gets inactivated. Now, that inactivation occurs when all of you women were at the 20-cell embryo stage. And so each one of your cells made a choice. Should it be this X or the other X that are turned off? So you're all variants, in a sense. And Jeremy Nathans wanted to study this. And he particularly wanted to know, are there any cells that don't do this? Are there any cells where it would you don't get XN activation. And so he made female mice. One of their X chromosomes allowed for the production of red fluorescent protein, and the other one allowed for the production of green fluorescent protein. He grew up the mice, and he looked at various tissues. And you can see that they're completely separated here. And this is the cornea epithelium, this is skin, but he looked at many other tissues to see this as well. Okay, so that's the GFP portion of the talk. Let me go on to trying to address the question of how do we make discoveries? How can we make these discoveries, or how can we promote them to make it more? I think, promising discovery. One thing, which I think this place does very nicely, and that Abraham Flexner promoted, he was the very first head, it was actually his idea, to put together the Princeton Institute of Advanced Study. This is where Gödel and where Einstein and all these incredible people were allowed to work, and this was his idea. So he's a pretty remarkable person. He said that the Institute for Advanced Study exists as a paradise for scholars who, like poets and musicians, have won the right to do as they please and who have accomplished most when enabled to do so. Now, there's several of these institutes around the world. I just want to mention 3 of them. One of them is Bell Labs, I already mentioned with Penzias and Wilson. And what I've listed under the Bell Labs are all of the Nobel Prize winners that did their work there. I was fortunate to do my postdoc at the MRC Laboratory of Molecular Biology in Cambridge, and here are all the people who did their work there that got a Nobel Prize in that. And finally, the Princeton Institute for Advanced Study, which at the time I originally made this slide, 41 of the 56 Fields Prize winners in mathematics had done work at this institute. So what makes these institutes, and I suspect here as well, so good at producing good science? Well, I think the following are the possibilities. I think the first thing is colleagues. It brings together people of diverse interests who are interested in each other and their work. There are the facilities that enable people to work. And similarly, there are the supplies there. When I was a postdoc at the Laboratory of Molecular Biology, I needed to have a control chemical that wasn't sold, but it was easily made. You had to shine ultraviolet light on another chemical, and that converted it. You purified it and converted it. And so I went down to the stockroom, and I went up to the head of the stockroom, and I said, I'm sorry, I need a UV lamp. And he looked at me all disgusted and just said, what wavelength? So that's the idea, to have everything available so that one can do one's work. But I think the other things are generally to have this support of people and this freedom to think, to be able to follow one's intuition or ideas and see how they work out without having to be forced to immediately get a result. Max Perutz, who was the head of the Laboratory of Molecular Biology and worked on the X-ray crystallography of hemoglobin, I think spent 16 or 17 years just working away at the whole thing before he had the ideas of how to actually do it. Just an amazing thing. And just that freedom to be able to do something really important, I think is quite wonderful. Okay. If many or most discoveries are accidents, how do we increase the number of accidents? How can we get more? One of them, I think, is do many experiments, right? If you only do one experiment, not likely that you're going to do it. But you do a lot of things, that's going to do it. I think another thing that's important, of course, is to do controls. We often say this. But I want to point out another thing. You shouldn't overdo it. Don't over-control the experiment. And I get this idea from a wonderful scientist. It was Seymour Benzer. He was interviewed once by the Genetics Society. He was talking about work that he had done at the beginning of his career with Max Delbruck on viruses. And they were studying viruses, and Seymour said that Max had a way of doing science which he referred to as the principle of limited sloppiness. And what this meant was, don't over-control. You can't control everything, don't even try, and maybe something unusual will happen. And the example from the virus field was they were growing virus in bacteria, and they wanted to see how many virus plaques would be produced overnight. And they did their experiments, very neat. They stacked everything up and came back the next day to discover that these two plates had a lot of virus and not so much, all the time. This surprised them until they realized that these top plates were the only ones that were receiving lights from the fluorescent lights above them. And it was the UV from those lamps that was inducing the production of the virus. If they had put all the plates all next to each other on the table, they wouldn't have seen any difference at all. So you can over-control. But I think that the really important thing is that we need to question and wonder more. I feel I need to question and wonder more, and I think there's two parts to this. The first one is, I think we should question what is right in front of us. And we should question our assumptions. I'm going to give you examples of both. I feel that there are questions all around us, and that we just often don't notice that these questions are around. So I'll give you some examples. The first one I want to give, it has to do with the fact that this was what I first worked on. My first scientific paper was based on studies of the major part of the body, and that was transparent. So I don't want anyone to say it out loud, but if you know what the major transparent part of the body is, raise your hand. And this is what always happens. It's a very small percentage of people that say this. But now I'm going to give you a hint. It's right in front of your eyes. Because it's the cornea. But that's a fascinating problem. How is that tissue transparent? And what maintains it? If you know anyone who's ever scratched their cornea, it becomes cloudy. So there's something controlling that. It actually turns out to be a transport system that keeps the hydration at the right level. But that's a fascinating problem, and you will agree with me that you have used this every day of your life. But very few people actually ever question this. And I've never met anyone, other than the people I went to work with when I did my research, who ever said, this is a problem I'd like to solve. So an everyday thing that we all see, but we all take for granted. Another example, and I should explain that in the next two examples, this one to some extent, but the next two examples are going to ruin your life. Because once you hear these, if you don't know them already, you're not going to be able to forget them. So I apologize ahead of that. But I want to talk about gravity and plant growth. There's a very old experiment that's done, I think it was old when Darwin did it, that if you look at a plant, of course, the root goes down, the shoot goes up. If you put a plant on its side, they turn so that the shoots still go up and the roots go down. So it's so-called positive and negative geotropism. So that's what everybody knows about, everyone's heard about this, but it turns out that that's not the only effect of gravity on plants. That in fact, all the other parts of the plants are also responding to gravity. And I'll give you some examples. This is a cypress tree, and you see that all of the branches are sticking straight up. Whereas in this fir tree, they're going up at an angle, and this one, they're parallel to the ground, and there are some mutants that don't seem to be able to respond, at least in some of their branches, and you get things like weeping walnut and weeping willow. You will never be able to go outside and look at trees now without thinking about this. That's the way it goes. Another example of this thing that we have seen every day of our lives, or almost all the time, but we never question, we never wonder about. This came to me when I was a graduate student teaching in a physiology course to medical students, and there was a professor that talked about the different types of muscle, of body, skeletal muscle. And he usually gave the talk towards the end of October, the beginning of November, and he would say, look, there are two types of skeletal muscle. One type, which we call the fast twitch muscles, they respond quickly, but not in a sustained way. They don't keep working forever because they go into oxygen debt. The other type of muscle works much longer. It's called a slow twitch. It can work for a while. There are sort of postural muscles that are important in this. And that needs an oxygen supply because it can't go into oxygen debt. And that oxygen supply is supplied by the molecule myoglobin, which, like hemoglobin, is a red pigmented protein. By this time, almost all the medical students were asleep in the lecture because this was not important. And he would wake them up to remind them that they were soon going to go home for Thanksgiving, or they were going to have turkey. Because if you look at a turkey or a chicken, the difference between these two, the dark meat and the white meat of these animals, is the difference between the fast twitch and the slow twitch muscle. Again, next time you eat chicken, sorry, you're going to think about this. I have a cousin, it actually is a second cousin, who is a professor of physics at Cambridge University in England. His name is Roger Goldstein. And Roger has made a career of looking at things that are all around him. So for example, one time he was trying to model a whole bunch of fibers flowing together. What happens if you had a bunch of fibers all touching one another? What shapes would they come? And he suddenly realized that he could use his model to explain the shape of a ponytail. He wrote this up, and that earned him an Ig Nobel Prize. If you know about this prize, if you don't know about this prize, look it up. They're fantastic. But he's done other things. One of the other questions he's addressed is, why are icicles shaped the way they are? Another question he addressed, our clothes are made up of very tiny fibers. So why don't they fall apart? And he has a model for that. And his most recent, we've just had an event in the United States where cicadas have all come out, the 17-year cicadas. And cicadas come out, they come out in groups. And so he was not trying to address why do they come out every 17 years, but he was trying to address the problem of why do they come out in groups. And he and his wife Adriana, who is a mathematician, modeled the emergence from the soil of these animals, so looking at how cicadas emerge, and what they found is they couldn't explain it unless they added one more thing. That was communication between the cicadas. And that seems to be there, and now I think people are testing that. So something around them all the time. I want to give one more example. For several years, I would go to this wonderful high school competition in the United States, or it's for the whole world, called ISIF, International Science and Engineering Fair. And I would go around and talk to people at the booths. And one day, I ran into Abigail Freed at her poster. And I asked her to explain the work, and she said something really strange. She introduced her scientific experiment by saying to me, look, the thing you should know is I'm a ballerina. Not exactly what you expect in the scientific discussion, but it turns out that she's not just she's a really good ballet dancer, and really good professional level dancers have a real problem. And that problem is that the cardboard soles that are in the dancing shoes go bad in one performance. So they perform, they got to buy a new pair of shoes, they got to buy a new pair of shoes after that. It's expensive. So Abigail decided that that was going to be her scientific experiment. She wanted to find a better sole for the ballet shoes. And she researched it and decided that she would work with a carbon composite material. This is her showing that one layer of the composite is too flexible. 5 layers are too stiff. But it turns out the 3 layers are the optimum thing, and they last for a month or more. So all of a sudden, she's made this wonderful discovery because she realized that there was a problem to solve. And I really liked it. I think she's terrific. I wish she would come to my lab. So I want to go into this other idea of questioning our assumptions. Now, some of you may know that there are these wonderful puzzles which are called matchstick puzzles, in which they give you a pattern in matchsticks. For example, here's one here, and they ask a question. Move 3 of the matchsticks and make 3 perfect squares. And you agonize, you move all the things around over and over again, and you finally decide, oh, wait a minute. I can move these 3, and now I have 3 perfect squares. And then you do another puzzle. And the puzzle is, here's a fish, drawn out in matches. Move just 3 of the matches and have the fish go in the other direction. And again, you play around with that, you move things around, and eventually you find that you can move those 3 and draw the fish out in this way. And that's fine, and it's really good, but then they really kill you. They give you this problem. Take 6 matchsticks and make 4 equilateral triangles. And as much as you move them around on the surface, it really doesn't work for the most part. And the problem is our assumptions, because the assumption is, why should we assume that it should be in 2 dimensions? If it's in 3 dimensions, you can make that structure. And now you have 4 equilateral triangles. But there's actually another solution to this. Because another assumption to this puzzle, which is never said in the question, is why do all the triangles have to be the same size? Because if they're not the same size, you can do this and wind up with 2 large triangles and 2 small ones. And so you have 4 triangles again. And then after you've said, okay, they fooled me once, they're never going to fool me again, they hit you with this problem. Make 3 matches into 6. I should tell you, you cannot break a match in 2. And you sit there and you look at it. Does anyone think they know the answer? Okay, well, the thing is, the assumption here, from everything I've told you, is that you should think about shapes. But that's the wrong assumption, because the way to make this 6 is to do this. So all of these assumptions were there because we sort of accepted them. And it's questioning these assumptions that are important. I have a friend, Ming Poo, who used to be in my department at Columbia, who's a spectacular neuroscientist. And one day, we were sitting around, and I can't remember who exactly asked Ming the question. I said, Ming, how do you get all of these incredibly good ideas for experiments? How do you do this? You do it repeatedly. And he said, I use the retrospective approach. What is the retrospective approach? He says, the first thing he does is he looks at a textbook. And actually, for him, the textbook was the second edition of Molecular Biology of the Cell. And he reads the textbook on whatever subject he's interested in and asks the question, why do they believe this? What is the basis of this work? And he goes and he looks it up, and he usually finds out somebody was guessing. They didn't really have good evidence for it. And he says, well, now with more modern techniques, I can really test this hypothesis. Either he's going to agree with it, in which case he can say, by more recent methods, we can also show and support this, or he's going to find that it was wrong. And after he tests it, if it's wrong, he can publish the correction, which he did many, many times. So it's a wonderful way of approaching science. Think about what we all assume and then question it and see if that introduces a new idea about how to do science. I've probably talked way over the time, so let me just finish with one of my favorite quotes about science. And this quote was by this person, who is my daughter. And in 2004, she was 12 years old. I had been elected to the National Academy, U.S. National Academy of Sciences, and they had an induction ceremony. Now, at the induction ceremony, if any of you have trouble sleeping, get a video of the induction ceremony because it is unbelievably dull. Because there's now about 100 people. The people in the audience are only there for one of them. All the other people they don't really care about. And each person gets up on the stage, shakes hands with the president of the academy, has a picture taken with the president of the academy, and they walk across the stage. There's a book that they're supposed to sign. They're told everybody in the academy has signed this book. And you say, I'm going to sign a book that Einstein signed. And then you find out they started it in 1970. But as you walk across the stage, they say something, a sentence or two, about why you were elected. So I'm doing that, and I'm realizing to myself, even though I love it that my wife and daughter are in the audience, that my 12-year-old daughter is going to be bored out of her mind. I have some experience with this. So I make my way down to where they're sitting, and I say to her, sweetie, I'm really happy that you and Mom are here. I like it that you're here to help me celebrate, but I want to apologize to you because you must really find this deadly. And my daughter looked at me, as she often looks at me, like I am the stupidest person on the planet, and says, Dad, don't you understand about these people? She had listened to those sentences. They were the first people in the world to see or do what they did. And that's one of the joys we have in science, is to be able to be the first person that sees something that no one else has been able to see before. And that's the real reward. It also means it explains something that almost every graduate student hates: failure. But if you're trying to be the first person in the world to look at something, you are going to fail most of the time.
Nice that you close your story with your daughter today, because today is the Father's Day here in Taiwan.
I forgot to tell her. I was going to do that. Oh.
All right. So I'm going to make an observation and a question. So I very much agree with you that about the usefulness of the useless. But the irony in your talk, my observation, is that you actually end up justifying the useless with their uses. It's just you end up justifying the useless with their uses. So their usefulness, they are actually useful, but they just came later. All right?
So, yes, I mean, the phrase is supposed to be provocative, but I think the idea is we don't know what insights we are going to get. I think, for example, if we take too narrow a view of what to study. GFP is a good example. The jellyfish are just not an important organism to study for human disease. Well, that's obviously wrong, but that idea that there are only certain organisms that we should be studying, I think that's incorrect. I think it's inappropriate. Often, there's a phrase that's used which is called using a model organism. I personally don't like that. I believe, although I have no proof for this whatsoever, that it is a phrase invented by doctors, because they want to say we are all working on human disease. These are models of human disease. I'm just being nasty, but that's how I think about it. But I know from the work in C. elegans that people have discovered things, and GFP is an example from the jellyfish, but in C. elegans, we've discovered genes that are important for cells to die, genes that are important for the timing of development, microRNAs, RNA interference, any number of things that are not models of anything. So I use a different word for these organisms because they're the ones that teach us what we should be looking at. I consider them not model organisms, but pioneer organisms. You're never really sure what those pioneers are going to show us, but it's always exciting.
Then comes my question. As a historian of science, I'm going to turn the retrospective approach on you, a scientist, right? So when you apply for grant, we always have to promise what's the use of our research, right? So if you look into your grant applications 40 years ago, what was your promise? And how correct were they?
Oh, they were amazing. So, and I'm not talking about me, I'm talking about the entire field. So, let me explain. When grant proposals have to be made, we have to talk about the significance. That is, are we doing an experiment that's worthwhile? That doesn't mean are we doing an experiment that will cure this disease or will make this product, but what's the importance about the work that we're doing? That's what the significance is. And yes, NIH grants always asked us to say what was the health relatedness of working with a worm, right, that we were doing. And we all said, well, there's this thing called evolution. So the genes must be pretty similar in worms and people, and if we study them in worms, we are going to get insights into the genes in people. And that's going to ultimately be important for health. And then when people started sequencing things, we found out we weren't lying. We actually were telling the truth. And so that's what we put in our grant proposals, because we profoundly believed in evolution. And this was the important aspect and what brought all these things together. In that case, it is a model. But just from our work on touch sensitivity in worms, we discovered new transcription factors that were then found everywhere. We found new types of channels, again, found everywhere. We found a new type of cholesterol binding protein that had a similarity to another protein in human kidneys that we worked on together with a wonderful collaborator. And so all of these things, we're finding this other connection. Are there some unique things? Yes. But we always had an argument in working with nematodes that was sort of we win both ways. And that was, if this gene that we're studying is found in people, then we have a great system to study that gene. If this gene is not found in people, that's a good target to get the parasites. And we can attack them. So we would win in both directions. We were very good at writing grants in my fields.
Okay, so this question over there.
So thank you for coming back. So I'm very inspired by this grant that you draw evolution relationship. But I want to bring a different aspect because now it could be a new era of synthetic biology. So how about we study those pioneering organisms so we can find something that humans don't have, but that knowledge can help humans to do something humans cannot do?
So, you know, I've often wondered whether anyone would like a green fluorescent tattoo. But I think they'd inject a dye in themselves and not transform their cells. So yes, that could be done. But I think GFP is an example of being able to learn about things. People have not studied in people directly, but studied in mice and other organisms how cancers metastasize, how cells become infected. There's a lot of studies that GFP has enabled because we can actually see the cells. And so I think there are a lot of things that we can learn. Look, the whole CRISPR-Cas9 revolution is changing everything. It's something from a bacterium, or from many bacteria. I think we've also seen in the basic research that probably has saved a good part of this audience by being able to enable the RNA vaccines to protect us from COVID. I think all of that basic research was really unbelievably important.
Okay, yes, please.
And I'm an undergraduate student working biology. And I just want to ask, what's your feeling after you finally get your expected results? And how do you look at your previous failure after you are succeeded?
How did I, what, the failure?
Look at your previous experiments failure after you are succeeded.
I'm not entirely. What? I'm going to try to answer that question. You mean, how did I cope with failure, or
what do you think about the failure after you
or after you succeed? When you succeed, you feel really good. You forget all about the failure. You know, it's oh yeah, that's in the past. There's something wonderful about having an idea and having it work, and you feel really good, and you tend to have amnesia. You don't remember these problems. You know, as I said, failure is a part of how we do science. Nobody can escape that. We hope that we do more things that will give fruit, but no one can be sure. But we're trying to do something new. And I think I personally get excited about the ideas. So I love it before we do the experiment because they're always right at that point. Then we do the experiment and I find that idea was no good and we got to do another one. But I like coming up with the ideas. And I would say the other thing I really like about laboratory work is that we don't do it alone. That it's a collaboration among people that are interested in the same problem and that we are working together. And I have to say, working with students and postdocs and colleagues is a real joy because you all have the same interest and you're working together on it. But that's me.
Okay. All right. Yeah, I think we...
There's one hand somewhere in the back
Someone
there.
at the end, maybe the last question there.
Okay, thank you for your speech. And as a medical student, I'm quite curious about, because we're medical students, and if our thoughts are often like, we want to cure something, or we want to invent or make some medicine or treatment for some disease. But do you think that we have to discard the previous thought and to, do you think we have the, do you think we need to discard the thought that we want to cure something and turn to be open-minded and accept wider fields of knowledge?
So absolutely not. So I think, at least in the United States, there was a really big push a few years ago for what was called translational research, taking what was learned in the laboratory and applying it to the bedside. That was the idea. And I think that that's not bad, but I think it's missing something. And what I think is missing is what I just mentioned before, the collaboration. I want you to be the best doctor you can be. And then I want you to talk to other people that are the best scientists and the best biologists and the best geneticists and the best people that are thinking about other areas so that together you can try to address the disease. So just as I said at the beginning, I'm not saying it should all be basic research and all applied. It's really working together. And I think that it is much better to be able to have people be open to this. I'm going to say a weird thing. I'm going to say a lot of weird things, but after I got the Nobel Prize, periodically I'd be on an airplane and I'd be seated next to somebody on the airplane that was a doctor. And I would talk to the doctor, and we're not talking a really old doctor, a fairly young doctor. And almost every time I did that, I found that they had no idea what GFP was. Absolutely no idea. I mean, it was on the front page of the New York Times, but they didn't read that. So to me, what that says is I'm not that arrogant that I want everybody in the world to know about my work. But I think it says to doctors that they should be thinking outside the standard curriculum, or in addition to the standard curriculum, to think about the advances that are being made in genetics and biology and everything, because they might have, they have the insights about the disease. If they also know how to connect up with others, as I just said, then I think some really interesting synergy can occur.
Okay, thanks.
I think we have already run out of time, but really like to thank Professor Chalfie for this wonderful talk. Wonderful, wonderful talk. Okay.
Thank you.